Welcome to Who's in STEM. I'm Ken Ono, your host, and the STEM advisor to the Provost and the Marvin Rosenblum Professor of Mathematics at UVA. Our goal is to evoke flights of imagination and wonder by showcasing the cornucopia of all that is STEM at UVA, the marvelous world of UVA science, technology, engineering, and mathematics. Now, I can't believe it. This is our 33rd episode. We definitely didn't know what we were doing when we started, but I hope you'd agree that we've kind of gotten the hang of it. But in our first few episodes, we had a news segment called Who's Making Discoveries. I keep my pulse on all things STEM at UVA, and I thought at the time when we were just beginning this show, I thought it would be a good idea to share timely stories. Now, in 2023, we had an episode with Francis Collins, the UVA alum who went on to direct the Human Genome Project. And he actually, by the way, won the National Medal of Science. In addition to my conversation with Francis, we opened with the following fun snippet from the news segment, Who's Making Discoveries? Please enjoy. As a spin on March Madness, a UVA health discovery has advanced to the Elite Eight in Stat Madness, an online tournament that crowns a significant biomedical achievement. The UVA discovery competing for the title is from the John Lukens lab. They've uncovered a molecule called a kinase that's responsible for orchestrating immune responses in Alzheimer's and multiple sclerosis. This finding sheds light on the toxic buildup of plaque in the brain that causes memory loss. Now, that was just over 1 year ago, and since then, John Lukens has certainly continued to innovate. John is UVA's Harrison Distinguished Teaching Professor of Neuroscience, and he is an affiliate faculty member of the Center for Brain Immunology and Glia. This past spring, he was named the inaugural director of the Harrison Family Translational Research Center on Alzheimer's and neurodegenerative diseases. Now, that's a mouthful, but it is a position and a charge that speaks to all of us. And as the conversation goes on, I'm sure I'll tell you about why it matters so much to me. But John, wow, when it rains, it pours. Welcome to Who's in STEM.
Thanks. It's great to be here.
So, John, we have a lot of very serious topics to talk about. Let's just dive in. I've had the opportunity to showcase UVA STEM science, stuff that's at the forefront of research in many different directions. And certainly, we've highlighted UVA research in the direction of Alzheimer's and neurodegenerative diseases. That's a big thing here at UVA. It's one of our grand challenges. So in an earlier episode, I had a conversation with Dr. Jaideep Kapoor and Dr. Sarah Kusinis, who co-lead UVA's Brain Institute. I've also had the pleasure of having renowned cell biologist Dr. George Bloom as a guest as well. And all three of them, and now you, my fourth guest in the field, think very deeply about the brain. But before we get into your work in detail and the institute you founded, refresh our memory. What do we mean when we're talking about neurodegenerative diseases?
So neurodegenerative diseases is just a broad term to describe a cohort of diseases that are associated with the death of neurons. So neurons control our basic movements, our senses. They help store our memories. So when you have loss of them in certain areas of the brain, you can have different phenotypes. So you could have your loss of memory, or you could have impaired movement. So in loss of memory, you're looking at things like Alzheimer's disease. And loss of movement, you're looking at things like ALS. And there are many common themes across neurodegenerative diseases. One is the buildup of neurotoxic material in the brain as we age. And this causes an inflammatory response and also cell death. And this seems to be a major culprit that could be targeted translationally to find cures for these disorders. And it's a really important issue of our time. Unfortunately, we really don't, at the moment, have disease-modifying treatments for the vast majority of these neurodegenerative diseases, which are greatly on the rise. It's an exciting time to be involved in this research, and there's a great opportunity to make a huge difference.
So some of these buildups, they're in the air everywhere. People talk about them, the amyloid plaques, right, that build up. Maybe you could talk a little bit more about that? For example, I know it's very difficult to even diagnose some of these neurodegenerative diseases. So when one studies neurodegenerative diseases as a broad category, what's involved in all of that?
Yeah, so it is a good point, and that's one of the major hurdles that we have at the moment is early diagnosis. Typically, people are presenting pretty far along in the disorders, so with some primary memory issues or issues controlling movement. So one of the big hurdles that we're going to face is identifying biomarkers that will enable us to modify the course of the disease. And there's been some movement there in terms of combining proteomics and genomics and precision medicine to identify combinations of things like amyloid beta, tau, and also synaptic damage markers in the CSF and in the blood. And so we're getting closer to having real biomarkers that could enable us to get in that very critical window to help.
But if you're a patient, or as we sit here today, you couldn't look at me and say, you're 10 years into a neurodegenerative disease, but I could be.
Yeah, and it can come on super quick, too. I mean, we're seeing it with some of infections in older people and also falls where everything's fine, and then a month or a couple weeks after a fall or kind of a viral infection, see people really turn a corner for the worse. So it can progress slowly over time, or it can come on pretty quickly.
Do you think there's a threshold beyond which if there's sufficient cell death or cell failure? I mean, is there redundancy built into the brain that protects us for a period of time as these diseases progress?
Yeah, the brain is, it sounds like an overstatement, but it's amazing in terms of its plasticity and how different areas of the brain can take over when there's loss in certain areas. So it's kind of amazing the plasticity that can be accomplished, not just in neurodegenerative disease, but in things like addiction and pain. You can rewire, and there is an opportunity, even if there is a lot of loss, to still regain some functioning that's missing. So, for example, there's individuals that have had issues with very serious depression and anxiety that was pretty much treatment resistant and, really didn't have hope for some of these individuals. And through various processes of cognitive therapy and others and, retraining some brain networks, we're seeing some improvement. The same thing can be said for PTSD, where there are these really large changes in terms of personality and mood, and the brain does have the capacity to rewire, and you can make an impact in that regard.
So I can add some personal experience. But about 15 years ago, my wife had a major seizure at night. It was the last day of school for the kids. We had just come back from a neighborhood party when my wife had the seizure. 4 hours later, we're at the hospital at the University Medical Center, and I was sitting down with a neurosurgeon who broke this news to me. He says, your wife has a brain tumor, but the initial scan suggests that it's not the most dangerous kind. It's not a glioblastoma, but a meningioma, which has very good margins, meaning that there was the prospect that you could excise it and recover and have a completely normal life, which is what happened. But with regard to the plasticity, I really want to share this story because it's a testament to the moment when I realized how amazing the human brain is. Within the period of 48 hours, we went from last day of school party to my wife's having brain surgery, and they removed this golf ball sized mass, which turned out to be in a very convenient location. And 2 days later, my wife can't write, she can't talk, she's like not there. And I remember meeting with the neurosurgeon thinking, my future it's changed overnight. He said, don't worry about this. This is the MRI. What you see is this cavity in the brain. This time next week, it will have closed up. And over the next couple of weeks, you'll discover that, well, maybe next week, Erica will learn how to take a step over a shoebox. And maybe 10 days from now, she can walk up a flight of stairs. And believe it or not, if everything goes well, she can go back to work in 6 weeks. And it actually all happened. And we were able to watch the progress because of MRI technology. I have those photos where cavity slowly closes up, and whatever happens, the human brain had this amazing ability to reconnect. She couldn't remember her name, then she could remember it. She couldn't write her signature, and then she could. And that was really kind of an amazing experience. So degenerative diseases come in many forms. right? The ones that I think we tend to be most afraid of include things that lead to cognitive decline and then like cancer. How would you compare cancer and how cancer progresses in a patient? And on the other hand, neurodegenerative diseases, which are quite insidious, right?
Yeah, there's an interesting comparison there. It's a lot of times there are overlaps in terms of, at least in terms of how the immune system can be leveraged to treat them. The brain has developed ways to limit immune activation, and that's what makes glioblastoma so difficult to treat. It does have this partially controlled setting where it limits interaction with the immune system. The interesting thing is there, like for cancer treatments of the brains, we're trying to boost immune activation. Whereas in many neurodegenerative diseases, we're trying to dampen down some of that activation so there isn't so much collateral damage. So interestingly, many of these pathways are shared between the two. So you can think of modulators that would both inhibit and also turn down by kind of studying the same kind of pathways or cell types.
I want to circle back to one of the main reasons we're here today. Congratulations. You're the founding director of the Harrison Center. Tell us about that. What are the neurodegenerative diseases that you want to study, and how's the work going?
Thanks. It's a real honor to be leading this new effort. It's desperately needed in the space of biomedical research, and it's just it's a real privilege to be in this position. It's going well. It's been a couple weeks. We're super excited for the new building. That's what we're working towards. We have a couple assistant professor faculty jobs out there.
What are you searching for?
So we're really looking for an interdisciplinary team. We want expertise in cell death, inflammation, neuronal connectivity, microglia, having this formed team with expertise in different areas. And that's really worked out well for us at UVA in the sense that we always have these checks and balances because you can't completely understand every facet of the disease. And so it helps to have different viewpoints and different expertise, whether it's at cellular level or at the circuit level, hoping to bring together an innovative group of people that just want to think outside the box to develop new treatments. And it's been really cool interacting with Evan Scott and hearing his vision and how the two new centers between Nanostar and us could interface is just exciting in terms of the possibilities. And leveraging their technology to identify and go after some of the molecules and cell types that we're discovering. There's been some incredible discoveries here at UVA over the last couple of years, and to have medicinal and nanotechnology in the same building to really jumpstart those treatments and discoveries of new compounds and ways to modify those pathways is just going to be awesome.
So this team that you're assembling, John, you mentioned that you want to bring together researchers that have different kinds of expertise that you can bring to bear. As the founding director, what's your expertise?
It's funny enough, I'm an immunologist by training. So I received my PhD from UVA. At the time, I was studying T cells and how they help eliminate viruses from the liver. That was my start in science. So I worked for Young Han in the Carter Immunology Center here at UVA. She's still here and a big supporter of me and our family. Yeah, it was interesting at the time because we were trying to basically rejuvenate immune responses. And at the time, there was this idea of T-cell exhaustion. Yeah, so T-cells are the cells that are activated to help eliminate cancer cells and virally and bacterially infected cells. So they come in multiple flavors. They're kind of the killer cells. So they say there's a tumor antigen, there's a viral antigen in that cell. We're going to lock and load and hit it. But what happens over time is the cells become functionally exhausted. So they still know that there's that virus or that tumor in that cell, but they lose kind of the payload and they become functionally exhausted. So this was thought to be a major driver of cancer development and progression. And so we and others were trying to figure out ways how we could boost or rejuvenate those T cell responses. And it was really cool because it was only a couple decades ago that this idea of cancer immunotherapy was born. And now there's multiple drugs that hit these pathways, these checkpoint inhibitors, that are saving people's lives. So, and at the time, it was kind of a crazy idea that didn't have that much support behind, but to see it blossom, and it's something that I always kind of talk about when I'm teaching in the classes, especially in the neuroimmunology classes that I teach in. We're at this early stage where we've recognized the importance of immunology, but we really haven't tested how efficient it could be to target the immune system neurological disease, and just even though I was only a small part of the cancer immunology checkpoint inhibitor. That was the rush that got me to where I am today. And it can happen in your lifetime. And it just takes a lot of energy and effort and enthusiasm to want to make a difference. And it definitely can happen.
Your career started here at UVA, as you just described, as an immunologist studying T cells. I'm not a biologist, but I don't think it takes much to recognize that you founding an institute to study neurodegenerative diseases sounds like wearing a different hat. So something must have happened along the way. So you talked about how the idea that one can combat and treat cancer by assisting the immune system. I'm guessing that in your research as an immunologist, you carried a little bit further. How does that work?
Yeah, so after I was at UVA, my wife and I moved to Memphis to work at St. Jude Children's Hospital. So St. Jude is the famous children's hospital that treats catastrophic children's cancers, but in addition, rare genetic disorders and also infectious disease. And it was just an incredible place to be for 6 years during my postdoc. One of the things there was don't just do science to do something incremental, but do it to make a huge difference. And it's kind of cool. I was there, at the 50-year anniversary, and we had this huge reflection and period. When we opened, what was the survival rates of this tumor in children, and what was the discovery that changed that number? And I think if you are creative and you think outside the box and you target multiple different systems, you can make a real impact on some of these disorders. So when I was there, I started working on multiple sclerosis. So multiple sclerosis is an immune-mediated neurological disease, and it's been one of the success stories. It's not a perfect success by any means, but one of the success stories that we have in terms of treatment of neurological and neurodegenerative diseases. In that, since it is an autoimmune disease, there was early buy-in to target the immune system. And we have multiple treatments that really slow the progression and enhance the quality of life of multiple sclerosis patients. And we don't have a cure yet, but we're moving a lot closer to that. And that experience of seeing how the immune system could alter the brain in the context of multiple sclerosis caused me to really figure out, the great thing about being in academic research is, you can follow your passions and your ideas. And it was looking at what was coming online, at least from some of the genetic studies and some of the imaging. It was clear that the immune system was involved in everything in the brain. But there was very little real deep dive into how that was potentially contributing. So I saw that as a real opportunity to dive in and kind of take my immune background and put it towards understanding neurological diseases.
So if I remember correctly, about 10 years ago, there was a huge discovery here at the University of Virginia. I know because as STEM advisor, I've been curating a list of major scientific discoveries. I think you probably know where I'm going with this. Science magazine, which is one of the most prestigious journals in science. I believe it was 2015. Correct me if I'm wrong, but I think in 2015 there was a major result coming out of the University of Virginia that Science called the breakthrough of the year. Tell us about that and how you ended up being part of this revolution.
That discovery was made by the great Yoni Kipnis, who was a major reason that I was brought to UVA and is still a huge supporter of me. And others. It was an incredible discovery, and Yoni is, he took a chance on a lot of us, bringing in an immunologist to our neuroscience department, but that's just kind of how he thinks. Basically, the discovery was that the brain has lymphatic vessels that are above the brain and just below the skull. The paradigm until 10 years ago was that the brain didn't have lymphatics, and this limited the interaction between the brain and the immune system. And what Yoni discovered and his team is right below the skull in this very thin layer, there were lymphatic vessels. And most people were missing it because when they do autopsies, they peel off the skull and they pull the brain out. But when they're peeling off the skull, they're also pretty much mangling the meninges and losing the lymphatics. And he identified this, and it was a massive discovery because it helped to break down the separation between the immune system and the brain. And since then, we've come to know through our work that this lymphatic drainage pathway is really important in protecting the brain from traumatic brain injury. And when it doesn't work as well, it can lead to things like tauopathies post-traumatic brain injury. And that's one of the projects that we're about to publish very soon. It's amazing how quickly that field's taken off since that initial discovery. And we've come to know that these lymphatic drainage vessels are really important in terms of eliminating neurotoxic compounds from the brain. So now we know that it helps to remove amyloid beta in Alzheimer's disease, alpha-synuclein in Parkinson's disease. And just yesterday, there was a really exciting paper that came out with early clinical studies from patients with Alzheimer's disease, where they did a surgical repair of this lymphatic vasculature. And the early reports are an amazing kind of benefit to these individuals that had Alzheimer's disease. And it's kind of gone viral. And interestingly, one of the major voices that they keyed in on for the perspective was the postdoc that discovered it at UVA. So it's been cool to see that unfold recently.
Well, John, that's amazing. In my field as a pure mathematician, a result that was proven 20 years ago is considered recent. But what you're describing obviously is quite groundbreaking. It represents a paradigm shift, and it allows, I guess, many more different kinds of scientists with different types of expertise to be significant participants. It sounds like Nobel Prize winning work.
Yeah, I would put my money on that. I think he's very deserving. He was doing neuroimmunology before it was cool. UVA was one of the first places to have a dedicated neuroimmunology center. And we're lucky to have been involved in the early stages of this field, and really grateful that UVA invested in neuroimmunology at that time.
Do you still communicate with Yoni? He's gone on. He's become world famous, and I believe he's at WashU in St. Louis. Are your team still collaborating?
Yeah. Yoni's like family. I was out to see him a couple of weeks ago. He's a very good mentor. Once he invests in you, he invests in you for life. So, my kids, when we drive out to Crozet and we go by his old house, they say, "Oh, there's Uncle Yoni's house." And
oh, wow.
It's been cool because he's developed this pipeline of new scientists, and seeing them, and some of them are trying to come back to UVA to start their own labs. So it's a really nice community. Since neuroimmunology is so new in the field of science, there was a really opportunity to establish this dynamic and open environment that encourages trainees to be really involved. And I love going to those meetings and seeing people like Yoni and Beth Stevens from Harvard, and the whole community is a lot of fun, and it's what science should be.
So now turning to the projects that really drive you, that you wake up every morning wondering about, the problems in the brain. Can you describe some of the specific research that the Harrison Center is going to be undertaking?
Yeah, so we're actively involved in trying to harness the immune system to help eliminate neurodegenerative diseases. We're studying right now is Alzheimer's disease. And it's interesting that rewind back 15, 20 years ago, it was that disease, everybody was just looking at neurons. That was the focus. But with advancements in genetic studies, we know that many of the genetic risk factors for Alzheimer's disease are only expressed by the immune cells of the brain, the microglia. These have the name of mini glue. Forever, I guess scientists just thought they were the glue that was holding the neurons together in the brain. But now we know they're the guardians that help to eliminate these neurotoxic compounds in almost every neurodegenerative disease that you can think of. And for whatever reason, over time, they become dysfunctional. And once they go off the rails, they just keep on perpetuating inflammation and cell death. That really is the thing that's the culprit that's really pushing it further and further down. So we're kind of using the same ideas that we had back 20 years ago to harness the immune system to fight cancer. We're trying to harness the immune system to recuperate those beneficial responses of the immune system. And back to a point that we had earlier, most biological systems are more plastic than we would think. Talking about the plasticity of the brain, many immune cells have incredible plasticity too. So there is a real opportunity to reprogram them for good.
So in analogy, or maybe directly related to your earlier work on multiple sclerosis, where you had the exhausted T cells, you had success in re-energizing or reactivating those T cells. Is the idea to replicate some of that success here?
Yeah, for sure. I mean, in terms of reactivating T cells, the one thing with glioblastoma is that for whatever reason, T cells don't function very well in brain tumors. They do a lot better in tumors outside of the brain. And there's many reasons that are thought to contribute to that. So that is a major crux of what people are going after, how to rejuvenate T cells to remove tumors. The other thing is the immune system's amazing. In addition to eliminating cells, it also plays this very important role in terms of being a protector, eliminating and basically engulfing and clearing away damage. And
this is the vasculature.
So there's both at the systems level with vasculature and lymphatics, but you can also think about it too on the cellular side of things. There's specialized cells that are like pretty much garbage men that go around the brain and kind of chew up garbage that could cause problems to the brain. Many of the same themes that people are hearing about in terms of the cancer space that were, a couple decades ahead of where we are in the neuroimmunology space are themes that are coming. So one of the things that listeners may have heard of are CAR T cells. And these are like engineered T cells that are like awesome fighters to remove tumors. Mm-hmm. People are working on CAR microglia. So they're genetically engineered microglia that have these beneficial roles that our microglia used to have earlier in life that help protect the brain. And there's a lot of emphasis, both academic and also in an industry, to develop these CAR microglia to target Alzheimer's disease and other neurodegenerative diseases.
So if in your family there's history of Alzheimer's, which is very common.
Yeah, there's genetic, environmental, it's really multifaceted, and I think it's like 1 in 6 or 1 in 7 over the age of 65.
Here's a question. With the therapies that you're hoping to develop, these synthetic microglia cells, for example, would the idea then be to somehow prophylactically prescribe these as therapies to someone who has a history or a high likelihood of developing Alzheimer's? Because as I understand, these diseases could take 10 to 20 years to develop.
Yeah, so that's definitely one of the strategies that we spend a lot of time thinking about. And we're working closely with Rich Price in the biomedical engineering department here at UVA. He has expertise in focused ultrasound and also genetic modification. And we are co-mentoring a really talented graduate student that is basically trying to do just that, but with genetic manipulation. So it's not actually a cell, but actually trying to genetically modify the microglia that are already in the brain and using focused ultrasound to target the specific regions and get it across that blood-brain barrier. So another thing that we think about, like prophylactically, getting back to this brain drainage discovery that was made at UVA, is we're studying it a lot in the context of traumatic brain injury. So obviously with traumatic brain injury, you have a much higher likelihood of developing tauopathies like CTE, which is what's become popularized because of NFL. What remains unknown is, why certain individuals are more prone to develop those issues later in life. And what our work has shown is that if you have a pre-existing impairment in this pathway and then you receive some head trauma, it makes things a lot worse. And on the other side of that if you rescue that after a TBI, it can limit the neurodegenerative disease later in life. So the thought would be, right now we're kind of saying like, oh, it's fine to go back to practice, or it's fine to go back to your military involvement because you seem cognitively intact. But a more empirical way to maybe address whether that individual is functionally safe to re-enter those types of situations would be to functionally understand if their lymphatics have returned to normal.
Is this something that can be tested?
So there have been some major advancements in terms of being able to image lymphatic functionality and structure in humans, and that's a very active area, and there's a lot of hope there. And there was a Parkinson's disease study in Nature Neuroscience a couple of years ago, really making that connection, correlating a cohort of individuals that had a lymphatic impairment to develop in Parkinson's disease later in life. So that is a hope. And it's interesting. Various people have shown it in various neurological diseases. But in our disease, there's this growth factor that you could either put on the skull or inject into the brain fluid, the CSF, that kind of is washing the brain, and it can boost the lymphatics pretty quickly and has a pretty profound effect in our experimental models. So there is hope that there could be a return, and I think looking at lymphatic function could really give you a sense of your neuronal health and your aging status. And almost thinking we go in for certain tests at certain periods of time. Why not look at your lymphatics too?
Yeah, that's exactly what I was thinking. Every time I go for my annual checkup, they take 2 vials of blood. I'm not sure I'd like the idea of them taking, a cc of my cerebral spinal fluid, but maybe we need that.
Yeah, there is a little bit of an aversion to taking CSF, but it provides an amazing window into the brain. At the molecular and cellular level, there are facets you can get from surveying the CSF fluid that you can't get from imaging. That being said, some of the imaging is getting really good at detecting the functionality and also the structural features of the lymphatics. So there are multiple ways that we could start screening these patients, probably in a way to take some of these treatments that can have a real effect and put them in play to really make a huge difference in their life.
So I'm sitting here wondering, the experiments that you perform, I assume you're performing them on mice. What are the test vectors, right?
There's various animal models that we're using. We also are looking at patient-derived cells. So we can basically reprogram stem cells to be microglia. And
in a petri dish?
You're
literally growing...
Yeah.
Okay.
Yeah, so one of the genetic risk factors that we're studying is a gene called INPP5D, and there's individuals that have this mutation and it's a gain of function. And you can actually take their cells and study how their microglia are dysfunctioning. And then, I was actually on a call earlier where they were doing just these studies where, seeing that dysfunctional microglia from that patient, basically drug screens or compound screens to see what it likes and what it doesn't like. Mm-hmm. So you could, target it better. There's various ways, but, a real power is, we removing certain proteins that we think are important and either eliminating them or adding them in higher amounts and saying, does modifying this one variable in a disease context, does it have an effect? And also looking at it both before disease and after the onset of disease to really say, do we have to hit it prophylactically before there's symptoms, or is there also a window where we could target it as the disease has already been diagnosed? We always approach it like that, just to really get a good sense of how good of a translational target it would be.
So, John, thank you very much for that very colorful and instructive kind of show and tell about what goes on in the lab. Yeah. Here at UVA, we are also a school, so I kind of want to circle back to the story about your training. You were mentored by Yoni Kipnis. Of course, here at UVA, we want to largely be measured by the success of our students. So now that you are running a center, you are the big name endowed professor, give us the behind-the-scenes look at the science and what it's like for you to mentor students in your lab.
Yeah, so, going back to the Stat Madness report that you highlighted earlier from a year ago, that was a study that was led 100% by a graduate student in the neuroscience graduate program, Hannah Ennerfeld. And she was just fearless in terms of pursuing that. The project actually started at the end of COVID, so we were had restrictions in the lab and it really had to communicate and collaborate with people in a different way. So we took up some collaborations with another group at Wisconsin, Tyler Euland, and it was incredible to see. And I was actually telling a student about that yesterday. From the initial working on that project to the submission of that paper to Cell, it was a year. And it just shows you when you catch on to something good and you put your heart and soul into it, and you almost become obsessed with wanting to understand to move it forward, how quickly science can move. And it was an amazing ride. And she was working closely with a really talented undergrad named Coco Hollywood, who, interestingly, is putting together a neuroimmunology symposium, a Gordon Research Conference, with a professor from Harvard, and she's now in his lab. And so actually we were talking about her earlier this week. But Hannah moved on. She's now a postdoc at Stanford, doing incredible work there. I'm actually going out there in a couple of months. She and another postdoc are hosting me for a talk. But we're hoping to get people like her back because they're amazing, and they're the type of people that make UVA better. And they're going to do much more incredible things than I ever did in my career, just because they're just a real joy to see succeed.
And the students you have in the lab now, the future Hannahs?
Yeah. Try and really create an environment that's fun. We have our third annual Spirit Week coming up. There's a lot of laughs. No question is a bad question. I think one of the things that we've really benefited from is having a lot of interaction with the undergrads. So every summer we have a whole cohort of undergrads, and they're always at meetings. I encourage them to ask questions, even when they're not prepared. And many of them come back and say, thank you for encouraging that, because it's definitely helped me in my PhD work now, wherever they are, or in medical school in terms of voicing their ideas, because that helps to move things forward. I'm co-mentoring a couple students, one with Bill Petrie on NeuroCovid, and then we have this translational work that Josh Samuels is working on with Rich Price. And you get to a point where they become your colleagues and collaborators, and it's like, that they're ready to move on when you're talking to them, just like you're talking to somebody like Yoni Kipnis or Bill Petrie.
Oh yeah, I totally know what you mean. Where you get to share, and in some cases maybe even relive the excitement that we had when we were graduate students, but to see that repeat itself over and over again. Yeah. In addition to the science, that's probably one of the most exciting things that a professor can, look forward to.
Yeah.
So you mentioned something. You said we have an upcoming Spirit Week. So I know what Spirit Week meant to me in high school and college. I don't want to repeat that, but what does Spirit Week mean in the Lukens lab?
Yeah. So I mean, the themes vary. It's a democracy. We actually had a vote today. The students, they have a good time with it. Everything from like Pajama Day, like is back in high school. They had a dress up like John Day.
John, that is awesome. So along those lines, the students that work with you, they must be super privileged to have you as a mentor. And so for students that might be listening, how can they get involved?
Yeah, so the easiest thing is just to email me, or, there's tons of information on our website, what individual people are working on in terms of projects. You can interact with them, email them. If people are interested to take a lab tour, or last week we had a first year join us for a lab meeting to see what it was all about. This is something we do all the time. We also are involved in this mini medical school program that happens at the hospital, where anybody from the community can come into the lab for a couple hours. And there are people that change courses and get inspired. There's actually a graduate student in our program that has come back to it as a second career because of a personal experience, and it's really inspiring to see that. And it doesn't matter if you have experience. I think the passion and the want to get better and to you make a difference is all you really need. And in terms of science, if an immunologist can do neuroscience, like there's no stopping anybody. I mean, that's the great thing about research. If you can put in the time and effort, and you'll get results and move the field forward.
Well, if you're listening, there you heard it. If you're invited to visit John Lukens' lab day, send him an email. And I think you can tell from this conversation, he is a very welcoming and easygoing scientist. Time is running short. I'd like to ask all of our guests questions that have to do with their origin story. I know we kind of talked about that going back to Yoni Kipnis, but I want to actually rewind the clock a little bit further. So when did you know you wanted to become a scientist, say an immunologist?
So I knew I wanted to be an immunologist my senior year of college. When I took immunology, I actually got a B in it. I didn't do that good in the class, but I loved it because immunology was fascinating to me because I got into science because I wanted to make a difference to help people that were suffering from an illness.
So you knew you wanted to go into medicine or biomedical research?
Yeah. I didn't think I had the chops or the wherewithal to deal with blood or surgery, but I knew I liked working with my hands to solve problems, and that's what research is. You have an idea and you try to use tools to answer it or disprove it. And, the thing that took me to immunology is it's hard to find a human disease where the immune system isn't the major player. You can think of autoimmune diseases like lupus, or you can think of cancer where the immune system can be harnessed to kill cancer cells. Obviously, infectious disease. And now we know that the immune system is a big player in terms of regulating most neurological diseases, too, everything from neurodevelopmental disorders to diseases later in life like neurodegenerative diseases. And that was really what drew me to immunology as a passion and research, and that's kind of how it started.
John, I think we can talk forever here, so we do have to cut this short. This is a fun moment, a joke, fun story from childhood. Tell us something that we could end this conversation with that tells us a little bit about John Lukens as a person, but not a scientist.
Maybe I'll tell you what I'm going to do after this. I'm going to go for a run. And there was a new Kendrick Lamar album that dropped a couple hours ago. I'm going to listen to that. Then I'm going to go home. I think there's a UVA game at 6 o'clock, St. John's. And then I'll hang out with my kids. We're playing indoor soccer in the house now, so we'll do that. And then that's a glimpse of the rest of my day. Maybe that helps.
Well, hey, you guys have been listening to Who's in STEM. We had to finish that way if you're going to follow UVA basketball. So, John, UVA is lucky to have you. This has been a super conversation. Good luck with the Harrison Institute Center that you'll be guiding. Good luck with your hiring. And I certainly am delighted to share that you are fulfilling President Ryan's mission for all of us here at UVA to be great and good in everything that we do. So I'm Ken Ono, STEM advisor to the provost and the Marvin Rosenblum Professor of Mathematics, and you've been listening to Who's in STEM.
Who's in STEM is a production of WTJU 91.1 FM and the Office of the Provost at the University of Virginia. Who's in STEM is produced by Catherine Kosseboom, Claire Curzon, Benjamin Larson, Mary Gardner-McGee, Katie Nichols, and Rhea Verma. Our music is composed and performed by Robert Schneider and John Ferguson of Apples in Stereo. Follow us on Facebook, Instagram, and Twitter. Listen and subscribe to Who's in STEM on Apple Podcasts, Spotify, or wherever you get your podcasts. We'll be back soon with another conversation about scientific and technological innovation at the university.